Yes.
Okay.
They cause infections which cause death worldwide. Okay, and in this picture that you see here, we see that all the continents actually are affected by this antimicrobial resistant pathogens. I'll just mention the scenario in India, where one child dies every 9 minutes from an infection caused by antibiotic resistant bacteria. Yes? Okay. What are antimicrobial peptides? Peptides are small peptides or proteins which are about 5 to 10 residues long, occur in all kinds of natural sources like invertebrates, plants, etc. They have got broad spectrum antimicrobial activity against fungi, bacteria, viruses, parasites, etc. They vary greatly in their sequence and structure. Okay, so there is no particular sequence or a particular structure for AMP. There can be a plethora of structures and sequences, and they have various modes of action, some of them being cell membrane disruption, anti-nuclease activity, anti-modulatory activity, etc. Now, though these antimicrobial peptides are very potent molecules, yet they have got some. Okay, so first, why are we interested in antimicrobial peptides? Okay, so now antibiotics, which are used regularly, they have got some problems in them. That is, they have specific intracellular targets, and because of this, development of resistance against the antibiotics is pretty fast. Okay, so the bacteria, they are very clever, or the microbes, and they develop some kind of a mutagenesis, which develops resistance against the drugs which are being used. Okay, but in the case of antimicrobial peptides, the development of resistance is very slow, if at all, or they never develop. Okay, this is mainly because of the fact that I just now told you that antimicrobial peptides have several modes of action. Okay, so they don't have one specific target. They can act on the microbe via various mechanisms. So due to that, development of resistance against them is very difficult. So that's how AMPs are potential therapeutic molecules. However, they have their own disadvantages. The naturally occurring AMPs are usually a little bit longer in sequences, and they are obtained in small quantities, and it's a tedious method to isolate them from natural sources. And because they are long, it's uneconomical for synthesis. They have poor systemic stability, or they have poor protease resistance, and they have high systemic toxicity, and there are peptide folding issues involved in the use of naturally occurring AMPs as therapeutic molecules. Hence, the synthetic organic chemists, they came up with designing
Yes, please. ও বাবা, রকিবউদ্দিন আমাদের ঝামেলা করছে। রকিবউদ্দিন আমাদের সাথে নতুন জয়েন করেছে। ঠিক আছে, দুবার তিনবার ঢুকছে আর বেরোচ্ছে, কি করে মাইক্রোফোন অন হয়ে যাচ্ছে আর কথা বলতে বলতে করছে।
in P2 and P3 was to change the positively charged amino acid residue lysine in P1 by arginine and histidine. While in P4, P5, P6, we did C-terminal amidation of the peptides, and we introduced a tryptophan instead of a leucine in the sequences P1, P2, P3. So P4, P5, and P6 have got one tryptophan each. And why we put a tryptophan was because tryptophan has got a very unique property. It has got an intrinsic fluorescence by which we can study, we can do a lot of studies with it, okay, biophysical experiments. So that was one thing. It would act as a reporter moiety, one. And number two, tryptophan has got a borderline hydrophobicity, due to which it has got a tendency of interacting with the lipids of the membrane. Okay, so that is reported in the literature. So that's why we chose tryptophan. So now looking at the antimicrobial activity, only peptides P4 and P5 were shown to have activity. P2, P3, and P6 were non-active, and P5 was more active than P4. And this was attributed to the presence of arginine in P5 instead of lysine in P4. Now, in the physiological conditions, if a molecule has to work as a therapeutic molecule, it would face a lot of salt concentration. Okay, so due to that, we checked the activity of these peptides in the presence of salt, and salt usually reduces the activity of the peptides, so which we also see in our case. But P4 and P5 still have activity against fungal strains like Candida albicans and Cryptococcus grubi. Here also we can clearly see that the MIC 50%, which is the minimum inhibitory concentration, is better for the P5 than P4, and the reason being the same. Okay. Next, if a molecule.
Just a minute, just a minute. Your presentation slide is not pinned up. Please pin the slide mode. You have two
What?
screens. One is yours and one is presentation.
Oh,
Just,
is it so?
yes, that's
Thank you
why
for letting me
so
know.
many
But
participants are asking that the presentation slides are not visible,
okay,
but they are not.
okay, just a moment. Hold on. I'll see how to fix the problem.
Okay, okay, no problem. It happens. Okay. There are two cells. You have one is presentation, another is only you. You are visible. Another screen is there. Okay, it's coming. It's coming. I am also sorry to disturb you. Okay, no problem. Okay, go ahead. Yes. Yes. অ্যাকচুয়ালি অনেকে তো সেই পেড ভার্সন সব ইউজ করে, তো অভি তো আমাকে ফোন করেছিল, বলছি আরে কি করবো, আমি বাইরে বলে দিলাম যে পেন্সিলের পেডটা ছিল, তো ফোন করে বলে দিলাম। আর ম্যাক্সিমাম আইআইটিগুলো তো তাই করে, ওগুলো নিতে আসে না, এই যে এর ক্ষেত্রেও তাই, সংস্কৃতির ক্ষেত্রেও তাই। আমেরিকাতে বহু আগে, আমিও থাকি তখন থেকে অনলাইন ঘর থেকেও বেটার।
microbe which is taken, and this is E. coli with P1. You remember the P1 peptide we started off with, which did not have antimicrobial activity. So you see that for the P1 peptide, nothing actually happened, okay? Now, till 100 micromolars. Now what we did was we incubated the peptides with P4 and P5, and these are concentrations of 1 times the minimum inhibitory concentration, twice and 5 times. And what we see is that both for P4 and P5, there is a total loss of the cellular morphology, there is deformation of the cellular membrane, there is leakage of the intracellular materials. Okay, so this conclusively proves that these peptides have membrane disrupting mode of action. Live cell NMR with Candida albicans, and we incubated the peptide P4 with Candida albicans, and we did an NMR. Okay, so here is the 0-minute profile or the NMR of the peptide. So you see the lines are sharp here to begin with. Now what happened that as we are incubating the peptide for longer time with the Candida albicans, all the lines, they're broadening. Okay, that is an indication that some interaction is occurring between these peptides and the microbe Candida albicans. Moreover, we see appearance of new peaks, you see, which were not there in the peptide. Okay, so this also proves that there is an efflux, okay, coming out of the intracellular metabolites from the cell upon disruption of the cell when we put the P4 peptide or incubate the P4 peptide with Candida albicans. So these peptides are membranolytic. And this is the control experiment, which shows that with P1 peptide, which did not have the antimicrobial activity, nothing incubate Candida albicans with the P1 peptide. You see, the peptides kind of look similar and it's the same. I wanted to see what is the secondary structure of these peptides that we have made, okay, when they are binding with the membrane. What is their secondary structure? So we did it with CD and with NMR. I'll show NMR in the next slide. So with the CD, what we see is that both in SDS and in DPC. So what is SDS? Sodium dodecyl sulfate, which is a surfactant, and it forms micelles, and these micelles are used as, again, microbial membrane mimetic. Why? Because microbial membranes are negatively charged and SDS is negatively charged. While DPC is another lipid, which is zwitterionic, and micelles, which are formed by DPC, they mimic the mammalian cells, okay, because the mammalian cells are zwitterionic in charge, okay. So what we see here is that all the peptides P4 and P5, they are random coil-like, both in the presence of SDS and
হ্যাঁ আমি দেখেছি ইউনিভার্সিটি স্টুডেন্ট ওই যে পান ছেলেটা ওকে জানি হ্যাঁ অনেকগুলোই আছে আমার নামগুলো মনে নেই কিন্তু আমার ইয়ে ফেসবুক স্টোরিতে আছে
and these are the MD snapshots of different peptides interacting with the SDS micelle. So this is P4, that's P5, this is P6, and this is a model 1, which is similar to a P1 peptide, only we have a tryptophan instead of a leucine, okay? But here we have a free carboxylic end, unlike the other peptides which have got a C-terminal amidation, okay? So what we find from here is that electrostatic interactions are the most important interactions that are present in between the peptide and the micelle, okay? And this interaction occurs between the positive side chains of lysine in the case of P4 and arginine in the case of P5 with the negatively charged head groups that is present in the SDS. So that is the driving force, okay? Even in the case of the real microbial cell, which is negatively charged, it is the electrostatic interaction which brings them close together and makes the peptide to interact with the cells in the first place. Now, secondly, there are hydrophobic interactions also between the hydrophobic side chains and these white parts shown here. These are the tail groups of SDS, okay, so parts which are present in the cell membrane. So there are presence of hydrophobic interactions as well, van der Waals interactions as well. Now, that explains why P4 and P5, they have activity while P6 doesn't, because P6, if you remember, it has histidine, and histidine amino acid residue has got a pKa at 6.5. So here we are in water at pH 7. So which means that at 7, the peptide P6 actually doesn't have any charges on its side chain. So here the histidines are non-charged amino acid residues. Due to that, there is a very small interaction, electrostatic component of interaction in between the peptide and the micelle. So it is inactive. While for the model 1, kind of represents our P2 and P3 kind of peptides. So there, because of the carboxylate group which is present there, so there is a negative charge here. That actually disfavors the interaction between the peptide and the SDS micelle, okay? So in short, if you have to summarize so far what we understood, membranolysis is the mechanism of action, electrostatics is the governing factor for peptide membrane interactions, and the secondary structure is not important for activity for such small peptides. Now, we wanted to improve on the activity of our peptides. We wanted to make more potent peptides. And what did we
শেষ হয়ে যাবে বলা যাবে না কত সময়